the signal from the axon hillock, which controls the firing of a neuron. Part of the neural membrane opens channels which allow for sodium ions to flood into the cell by the concentration gradient, which is an area of higher concentration to an area of lower concentration. This process, also known as our sodium-potassium pump, will cause a rapid increase in the positive charge of the nerve fiber. Once the threshold of excitation is reached, or we move from a negative 70 millivolt charge when we are resting to a negative 55 millivolt charge through this depolarization, the neuron will fire an action potential, which is an electrical signal down the axon, which is a long portion of a neural cell, almost like a tail. Every skeletal muscle fiber is innervated with an axon. So at a muscle, the axon gives branches to many muscle fibers, but each fiber gets only one branch. A motor neuron and the fibers it innervates together are called a motor unit. This motor unit then converts an electrical impulse to a chemical impulse through a series of complicated events that we've depicted in a number of beautiful drawings here. An axon end is bulbous, the distal portion of the neuron, where it comes close to but does not actually meet a muscle fiber. The axon is considered the signaling portion of the neuromuscular junction, or also the presynaptic terminal. There's a few interchangeable words here and throughout this. The axon is also surrounded by a multitude of positive calcium ions. There are three distinct portions at this junction. There is the bulbous end of the axon, the synapse or the synaptic cleft, which is a gap in between the axon and the motor end plate, and is filled with extracellular fluid and a multitude of sodium ions, and the motor end plate, which is on the muscle fiber itself. The motor end plate is comprised of a very convoluted surface that is the receiving end of the neuromuscular junction. A very convoluted surface makes for more surface area, and greater surface area is great for our bodies when we need to absorb something. So now that we've cleared up some of the anatomy of a motor end unit, we need to clarify the transition from an electrical impulse to a chemical impulse across the synaptic cleft. At the synaptic knob, there are voltage-gated calcium channels. Inside this knob are many mitochondria and synaptic vesicles. They're tiny transportation vehicles, like dump trucks, filled with a chemical signal called acetylcholine. When an action potential, which is still electrical at this point, reaches the synapse, these channels open, causing calcium ions to flow into the neural cell. All of this calcium flooding into the cell pushes the acetylcholine-filled synaptic vesicles to the distal end of the neural cell, and the synaptic vesicles fuse to the cell membrane and dump the acetylcholine into the cleft, or the synapse, through exocytosis. With a chemical signal now ready and able for the muscle fiber itself to use in the synaptic cleft, the motor end molecules. So don't forget, like I mentioned before, we have sodium ions here in the cleft as well, and not by accident. There are acetylcholine receptors on the receiving or proximal motor end plate that are in conjunction with sodium ion channels. When the acetylcholine binds to its acetylcholine receptor site, it opens these sodium ion channels and allows for sodium to flood across the cell membrane, also called the sarcolemma of the muscle cell. Before we deal with the muscle fibers, we need to address the acetylcholine that's still sort of hanging out in the synaptic cleft. It needs to be broken down so that sodium won't continually flood the muscles, which would result in constant muscle contractions. There's an enzyme in there called acetylcholinesterase, which breaks down acetylcholine into acetic acid and choline. The choline is then reabsorbed back into the presynaptic terminal and combined with more acetic acid to form more acetylcholine. That newly formed acetylcholine enters the synaptic vesicles and awaits another electrical action potential to contract a muscle. Now that a muscle fiber has received the flooding of sodium from the chemical signal it received across the sarcolemma, we can begin to understand what Now that a muscle fiber has received the flooding of sodium from the chemical signal it received across the sarcolemma, we can begin to understand what happens to a muscle fiber after it depolarizes. The sarcolemma, or the cell membrane itself, is a very complex structure consisting not only of the phospholipid bilayer, but of many penetrating tubes that are transversely positioned into the cell, conveniently named T-tubules. These T-tubules allow depolarization to happen quickly and penetrate the cell's cytoplasm. Here in a muscle cell, it's called sarcoplasm. Just beneath the sarcolemma lies a web-like structure called the sarcoplasmic reticulum. It fits almost like a sleeve around the entire muscle fiber and contains 10,000 times more calcium than the rest of the body in a binding protein called calsequestrin, named conveniently for the service it provides to sequester calcium. Most of this is contained in the terminal cisterna at the end of the sarcoplasmic reticulum. So calcium is released by the sarcoplasmic reticulum during a muscle contraction and reabsorbed by the sarcoplasmic reticulum during relaxation. The flood of calcium triggers the sliding filament mechanism. This is an explanation for how muscles produce force or shorten or contract. There are two types of fibers, or myofilaments on this level, and I'll get to that in just a second. There are two types of muscle fibers in regards to girth, I guess. There is actin, our thin filament fibers, and myosin, our thick filament fibers that also support a hinged or pivoting globular protein head. So when you break down a muscle, we have a whole muscle, which we broke into cross sections and bundle by bundle. A bundle of fascicles surrounded by perimysium, which is connective tissue, make up a whole muscle. Fascicles are made of many muscle fibers surrounded by endomysium, which is also connective tissue. Those muscle fibers are made of myofilaments or myofibrils, which is where the sliding filament mechanism begins. Each muscle fiber is separated into neat little sections by two Z discs. Here we have a perfect display of what a sarcomere looks like at this level. The Z disc is a continuation or a seat, so to speak, of our thin filaments, which is actin. So when a muscle contracts, the Z discs move closer together. The A zone, where the myosin or thick filaments are stationary and overlap the thin filaments, thus creating the striated appearance of skeletal muscle. The A band remains relatively constant in length during contraction, while other regions of the sarcomere are shortened. The I band, containing actin, changes its length along with the sarcomere. Because actin is tethered to Z discs, any shortening of the actin filament ends in a shortening of the sarcomere, and thus the muscle. The H zone sits right in the middle of the A zone, and this is the portion where the actin and the myosin do not overlap. Also, the H zone is not visible when the muscle is fully contracted. When the calcium floods the sarcomere, it binds to a protein called troponin. Troponin, during rest, sits like a lock on a long, thin rod-like protein structure called tropomyosin, and this wraps around the actin filaments and covers the binding sites on the actin. When the calcium binds to the troponin, it unlocks it and moves. When the calcium floods the sarcomere, it binds to a protein called troponin. Troponin, during rest, sits like a lock on a long, thin rod-like protein structure called tropomyosin, and this wraps around the actin filaments and covers the binding sites on the actin. When the calcium binds to the troponin, it unlocks it and moves the tropomyosin to expose the binding sites for the myosin head on the actin. The hinged myosin heads have an ADP molecule and a phosphate molecule attached from a previous cycle of movement and are in a high energy state. The myosin heads leap to form a cross bridge between the exposed binding site on the actin and the myosin filament. Once the cross bridge is formed, the phosphate molecule is released and energy stored in the myosin head actually moves the bound actin across the myosin filament. This movement is called the power stroke. The ADP is released as the myosin head moves. The cross-bridge bond is broken when another ATP molecule binds to the myosin head, and then the attached ATP is broken down into ADP and a phosphate and awaits another chemical signal. This entire process makes for a tug-of-war motion between the movable actin attached to the Z disc and the central and stationary myosin. Once the cycle is complete, calcium is reabsorbed back into the sarcoplasmic reticulum, which allows the tropomyosin to cover the binding site on actin and relock the troponin.